All right, everybody, we're going to start our next session. So we're on session 3 of 4. We've already had some really good talks this morning that introduced where the Delta RMP has been, the Delta Nutrient Research Plan, the various studies from USGS, Regional Senn, all of that work, the cyanobacteria, some updates with that research. So we're going to jump into our modeling. We've got 3 presentations. We're going to have David Senn about the Delta-Suisun biogeochemical model. We have Domagalski, who's going to talk about the SPARROW model from USGS. And then we have Zhenlin and Zhang, who's going to talk about our last biogeochemical model, the modeling delta water quality using coupled hydrodynamics and biogeochemical models from DWR. And so with this session, we're going to hear a lot about what was talked about from our first couple of sessions about models and their importance for predicting management effectiveness, for understanding some of the different factors that feed into the nutrient cycling. And so this is a really good place then, before we hit our 4th session, to get an update on these 3 efforts. Okay, so I'm going to hand it off to Dave Senn.
Thank you. Okay, and I'm going to try to get you on share here. So. You see the presentation on the front?
There we go.
Great, thanks. So I'm going to be talking about the development and application of a hydrodynamic biogeochemical model for the San Francisco Estuary. Presenting on behalf of my collaborators at SFEI and Euclid. I'm going to start off with background and science questions, and then just a really brief overview of model details. And one of the reasons for that is because we've already presented this a number of times to the Delta RMP. I know there's a lot of overlap here, but I also have things at the end to talk more about potential applications and limitations. So I'm going to go through the details and then jump to the results and interpretations, and come back at the end to talking about model skill and some of the key limitations and areas for development. And so, first of all, I wanted to acknowledge some funding from a variety of sources, as Dylan noted earlier, and others as well, that there's been a lot of funding sources that have come together to support this work, from the State Water Board, Delta RMP, Delta Science Program, Regional Senn, Central Contra Costa Sanitation District, and the San Francisco Bay Nutrient Management Strategy. And one of the things that was great about some of this work was that we were able to also very early on agree that the various funding sources were okay with the idea of shared products. And so we were able to move forward an effort that was this big in small pieces, where probably no one would have wanted to fund their own little piece of development, but they were all interested in having the overall package done. And lastly, I wanted to note several collaborators over several years and projects, starting off with Zhenlin Zhang, who's here, who really was the person who spearheaded this initial work, our initial work in the Delta. The USGS team, the BGC team, Lisa Lucas from USGS, and other collaborators, including Janice Cook, who really helped secure these funds, as well as help point us in the right management direction. So one of the things I'm hoping to cover, either implicitly or hopefully explicitly at times, is the goal, the direct linkages to the DNRP. And I think so, Melissa, one of the reasons that I was late getting this presentation to you was I was reading through all the questions here, and then also the color scheme. The color scheme was really tricky. From the purple represents aspirational or could go in that direction. The red means yes, we're definitely trying to do that. So it's all here.
Perfect.
Great, thanks.
Done.
So instead of going through that long list of questions, I'm going to hopefully go through this shorter list of questions, which is related to this report that we came out with last year, 2021, Nutrients in the Northern San Francisco Estuary, Transport Cycling and Forecasted Changes After Nutrient Load Reduction. And so the questions that we were exploring in this report were, how do nutrient concentrations vary spatially and seasonally throughout the estuary, the northern part of the estuary, Suisun Bay and the Delta? What physical and biogeochemical processes or mechanisms regulate nutrient transport and fate, and ultimately the concentrations or nutrient availability in the system? What nutrient sources contribute to regions that are impacted by HABs or invasive aquatic vegetation? really careful in the choice of words there. We're not actually trying to simulate the haves or simulate the invasive aquatic vegetation at this point, but more simulate the sources of nutrients to those regions. What factors influence those fluxes to those regions? And how will the regional sand upgrade influence nutrient fluxes, transformation concentrations, and delivery to downstream systems? So I wanted to also put this in a bit of context for the overall program that I spend most of our time, or our team spends most of its time working on, and that's the San Francisco Bay Nutrient Management Strategy. And we have 3 model domains used for different regional applications: a lower South Bay focused, ultra high resolution model for getting into the sloughs and creeks and salt pond connections, a core full bay model, and then a Delta-Suisun focus where it's a slightly different grid for the open bay part, but includes the coastal ocean. We're also exploring impacts on the coastal ocean of nutrients leaving San Francisco Bay and going out to the coastal zone, collaborating with researchers at SCORP, UCLA, and UC Santa Cruz. And so there's something I'll just leave in here. I won't go into any of this, but just to give you a sense of some of the various work that's there. And there's also at the very end of the slideshow links to some more details about the various applications. So we first got started on this, and it was mentioned earlier, I forget exactly whose presentation mentioned it. Oh, I think it was Tim mentioned the mass balance of the delta. We were needing to understand nutrient loads coming from the delta into San Francisco Bay and into Suisun Bay. And this is a graphic from one of those early reports. October through September, kilograms per day nitrogen of both the loads directly to Suisun Bay from PUOTWs discharging directly to Suisun Bay, and then the loads coming from the delta into the system. And so you can see the really strong seasonal variation, including dominance of nitrogen loads coming from the delta into Suisun Bay. And so a key question here, in order for us to understand not only conditions in Suisun Bay, but also elsewhere down gradient in the system, how do delta to Suisun loads vary seasonally and interannually? How do delta Suisun loads change in response to the regional sand upgrade? And this was something that, we can use some empirical approach, make fluxes into the system on observational data, but once you actually fundamentally change the system, you can no longer rely on those empirical approaches. And so we knew we needed to get ourselves up into the delta somewhat, at least, to be able to simulate those fluxes coming out into the rest of the system. In addition, it's a fascinating and important place to work. And so I think we probably went a little bit deeper than just the needs of what we needed to do for the bay work. Well, Dylan mentioned this earlier. I had an opportunity to work with Tamara and others on this report as well, changing nutrient inputs to northern San Francisco estuary. What are the potential changes that could happen to the system in response to the regional sand upgrade? And so among the many responses and the many tortuous diagrams that we managed to come up with through this report, the examples of potential changes were decreased phytoplankton production due to nutrient limitation of phytoplankton growth rates, decreased HAB frequency severity, decreased invasive aquatic vegetation, or density or aerial coverage. But one of the key things is, if you want to be able to know whether all these things are things you could imagine or everyone could come up with when you're thinking about reducing nutrient loads. But a really key thing is, in order for that to actually be realized, you have to be looking in, or that issue that you're trying to explore needs to be inside the area that's influenced by the effluent plume. And so, for example, if you're interested in nutrient-related response B, there needs to at least be some overlap with that effluent plume, or that zone of influence, which we started calling it. And that zone of influence decreases, or the intensity of that influence decreases with increasing distance or travel time. And so we came up with this approach. This was something that Zhenlin and I worked on to develop this concept of a zone of influence going from maximal to minimal. And this was intended to look like a contour plot. It looks like some sort of 1970s bird flight or a dove or something like that. But you get the idea. It's a plume that then branches out and spreads out into different parts of the delta. Maximal, this is meant to be summer. And this is for the dissolved inorganic nitrogen. So imagine, like, as you get further away from, as you get into these lighter colors, you start to be in a place where the upgrade would be felt less. Exactly where that boundary is and exactly how much less will depend a lot on the conditions in the system. And so something we need to think about then is these issues that we're concerned about in terms of impacts. In particular, for example, the severity of cyanohabs. This is another really rough mock-up of where it's most intense, and this was nicely illustrated by ongoing data collection by Keith and Ellen today. This is a region in the southern delta or the interior delta where it's the most severe conditions related to HABs, and similarly, floating in invasive aquatic vegetation, water hyacinth or agaricus. These are all things that are quite distant from at least the core of this zone of influence, and so in addition to one of the things we needed to understand is how far is this zone of influence felt, and what magnitude is this zone of influence felt? And that's one of the key things that we were exploring. In addition, as I noted earlier, we're not simulating right now invasive aquatic vegetation. We're not currently simulating cyanohabs. But instead, what we decided to pursue was something that did seem tractable, which is what is the flux of nitrogen into these regions delivered across coming from not just the concentrations, because concentrations could be deceiving. You could have low concentrations, and this is something we've been encountering. USGS and SFEI have been working extensively on the bloom in San Francisco Bay in recent weeks and month and a half. And one of the things we're finding is that nitrogen levels are continuously low throughout this bloom that keeps spreading. But it still keeps spreading and growing. And so nitrogen levels are low. It's getting the nitrogen from somewhere, kind of a steady state uptake of nitrogen at the same rate that it's being delivered to the system. And so understanding the fluxes instead of just the concentrations is one of the key things we're exploring. Similarly with the floating aquatic vegetation and submerged aquatic vegetation. Thanks. So really briefly, the model setup. The model domain extends out to the delta. 70,000 horizontal grid cells, 10 vertical layers. You can see a bit of the the detail on the grid, including Frank's track there. And the model approach that we're using, or the model tools we're using, the Deltaris Delft Flexible Mesh, calibration, validation of water year 2016, and there's a link in here to find that report. Biogeochemistry, Deltaris DelWalk, with calibration and validation for water years 2016 and water year and 2011, where we were aiming to two very distinct years in terms of their hydrological forcings or wet-dry. And so we thought, looking across those 2 years, to come up with the best possible calibration, the optimal calibration across that time. We're also at least pursuing this in an open source community modeling mindset. I don't want to say that we're actually, like, we don't have this thing up on a website for people to download, et cetera, yet. That's the approach we've been taking, and we recently put a proposal into EPA to support some of that documentation and posting and sharing. And many of the investments that we put into that, especially over the last couple of years, has been in an empirically driven light attenuation field using the 40, 50 turbidity sensors throughout the bay. So we have daily average spatially interpolated light fields, dynamic benthic grazers, 2 different grazers, and dynamic zooplankton, sensitivity analyses for calibration, sediment diagenesis. And then in addition, mass balances approaches both to assessing model performance, but also assessing interpretations, or in terms of developing our interpretation. So not just looking at, say, the concentrations, but very much looking at the rates and the fluxes between compartments of the system. This is a quick glimpse, and I won't dwell on this, but you can see this in the report. Dissolved inorganic nitrogen, milligrams per liter. Some of the stations, the IEP, EMP sites, D28, D26 are in the interior delta or the central delta. And then there's the other stations in Suisun Bay and along the Sacramento River. One thing I want to point out is this is like, probably with a lot of modeling efforts, we're constantly straddling the report we just finished with the new calibration that we're coming up with, and not quite always caught up with the analyses and interpretation. So this is the calibration for our newest implementation. The interpretations I'm going to be showing you are from a slightly older version, but the nutrient fits, or the goodness with nutrients, are the same, and we don't expect that it will change the interpretations much at all. So one of the things that, in addition, that we did was would you click once more, please? Thanks. So we ran the model in high resolution throughout the system, but then we set up these control volumes throughout, and we set up these control volumes, regional control volumes, to do mass balances, like fluxes coming into a particular region, transformations within that region, and then exports to other parts of the system. And so one of the things that I'll show you next is sort of we tried to slice these up both to be defined based on other criteria that including the water board proposed. And then in addition, slice them up so that there's kind of a limited number of connections between these different control volumes. You can kind of have a discrete in or a couple discrete ins and a couple discrete outs. Thank you. And so this is a blow up of one of these locations, the central delta. This is where this includes Frank's Tract. And this is now we're getting into the area where there are problems with invasive aquatic vegetation. There are periodic harmful algal blooms occurring. And so what I'm going to be showing you next is a plot that will show the mass fluxes over the course of this year coming into the system. But could you go back one? Oh, thanks. So this is now showing, there's a lot of arrows here. And so but just look at these first, and just the ones coming in. The ones that I realized afterwards that I should have color coded this to match the diagram, but I already did so much color coding earlier that I was tired of it. So but now could you click ahead, please? Yeah, thanks. So now this is showing import of dissolved inorganic nitrogen to the central delta. And so I'm going to take a moment to explain this as opposed to just blowing through it. So this is going from October 1st through August 31st. October 1st of 2015 through September of August 31st of 2016, megagrams of dissolved inorganic nitrogen per day. This is a stacked plot. So what's the total, if you just look at the total, like for example, the 15 megagrams per day in roughly February, that's adding all of the different sources up coming into the system. And so the red is coming from the McCulloughney tidal transition zone, so coming from north to south. Import from the Western Delta is this arrow coming in, so coming out of, blanking on the name, 3 Mile Slough, I believe. And then heading out this way is what I'll show you in a subsequent plot, leaving the Delta. And then there's also potential for nitrogen to be coming in from the San Joaquin tidal transition zone. And so the other important feature of this graphic is the gray area in the back, if you can see that, is where it's white, that means that the delta cross channel is open. Where it's gray, the delta cross channel is closed. And so one of the things that you can see is how the red coming in from the McCulloughney tidal transition zone increases and decreases in response to the delta cross channel being open and closed. I get no surprise from a hydrology standpoint, right? But this isn't water, this is nitrogen. And so in the report, there's a graphic showing this more clearly, but when the delta cross channel is open, roughly 50% to 60% of the nitrogen is coming through the delta cross channel. The remainder is coming through Georgiana Slough into the system. So, all right, so when the delta cross channel opens in June, sporadically, you get roughly roughly a 50%, somewhere about a doubling almost of nitrogen coming out of the Sacramento River, coming into the central delta via the McCulloughney tidal transition zone. We also ran tracers, point source tracers, or what's the right word for that? Conservative tracers to give you an indication of where the water was coming from. And so what this is showing here is a plot with the same sort of information about the delta cross channel being open and closed, but the percentage of the Sacramento River, the black line is showing the percentage of Sacramento River on average within the central delta polygon. So as the delta cross channel opens up, the percentage of Sacramento River water in this region on average goes from about 40% to 80% by the end of August. So roughly a 50% increase in DIN loads to the central delta coinciding with the delta cross channel opening in June 2016. New DIN loads primarily from the Sacramento River, and percent SAC increases from 40 to 70, roughly 80 by end of August. So where does the DIN leave to? So this isn't filling up like a big bathtub, water is moving in and then moving out to other regions. And so this is now showing the export along, so the arrow is leaving the system. So the arrow to the right going out, going to the west is export to the Western Delta, and the purple is export to the Delta transition zone. So down to the south, down to where the pumping exports occur. And so one of the things you can see again is that as the, well, actually, would you mind clicking that, please? Thanks. So June through August 2016, the Delta cross channel was open. 30 to 95% of DIN exports from Central Delta flow to South Delta transition zone. So as you go out later in the year, most of it is moving south into the Southern Delta. So now asking, like, the question is, DIN sources to another region, the South Delta transition zone, another area that's influenced by invasive aquatic vegetation. And so please, thanks. So now this is showing the import of DIN to the South Delta transition zone, and also the sources of where that nitrogen is coming from. So the blue is showing import from the South Delta, so coming from the south in this direction, and the green is showing the import from the Central Delta during this time. This is now showing where it's going, and this kind of reminds me of The Little Prince, it's like the snake that swallowed a hat or something like that. But this is one of the things you can see as the gates open in the Delta cross channel, and as the pumping goes up, it's drawing water across the system and down into this part of the system. Of course, that's why it was designed, was to pull water in that way, but it's also pulling the nitrogen loads in that way as well. And so roughly 100% of the DIN inflow is coming from the Central Delta from May through August 2016. So during the warmest times of the year, when the issues like highest growth of aquatic plants as well as highest potential for growth of cyanohabs, there's delivery of nitrogen coming from, or this model simulation is suggesting that delivery of the nitrogen is coming from the Sacramento River. So next question, how will the regional sand upgrade influence nutrient fluxes, transformations, and concentrations, and nutrient delivery to downstream systems? And so what we did to explore this was we took the same water year 2016, all the same forcings, and instead of using the pre-upgrade loads, we upgraded the system in water year 2016 and ran the model during that time. And so now what this is showing is for a variety of different stations, but fluxes into Suisun Bay and also concentrations of nitrogen from one representative station, an EMP station in Suisun Bay. So roughly a 30% DIN load decrease to Suisun Bay and the rest of San Francisco Bay as a result of the upgrade. And that's roughly year round. It's hard to tell from that graphic there, but you can see the pink is the loads from the wastewater treatment plants direct to the system. And then you can see that the blue line is DIN imports or influxes pre-upgrade. The green is DIN imports post-upgrade. And then now showing as well at some of these more interior Delta stations, D19, D26, D28. One more time, thanks. Roughly a 50% decrease in DIN concentrations in the Central Delta as a result of the upgrade, as well as 30% decrease at least the one station where we have we're showing here D28A in the northern part of the South Delta transition zone. And so by no means are we trying to suggest that these are homogeneous blocks and that the source of the nitrogen is the same and that the changes would be the same due to the upgrades. But representatively, we're just aiming to show here with the stations that people think of most, especially when we're validating the model, like how these changes might affect concentrations in different parts of the system. So what physical and biogeochemical processes or mechanisms regulate nutrient transport? Well, at least to the interior delta, the operation of the delta cross channel, as well as the pumping, at least this simulation suggests, has a major role in inducing flow into the system, as well as bringing the nutrients associated with that flow into the interior part of the delta. I remember the first time this kind of clicked for me, it was a simulation that Zhenlin had run, and we were looking at pre and post upgrades several years ago. And it's like, oh my gosh, something just happened in mid-July that all of a sudden we see this change that I was like, there's something wrong with the simulation, did something, and no, it was actually a breakthrough of after the Delta cross channel had opened up and you could see it in a certain location. And so what nutrient sources to regions that are impacted by HABs or invasive aquatic vegetations, at least pre-upgrade, this simulation suggests that even though it's quite distant from the Sacramento River, that the areas that are impacted by HABs and aquatic vegetation are within the zone of influence to the tune of roughly 50% of their concentration. And so one of the things I guess I should really make sure that I'm attenuating my statements on this, because like I see this as very much a quantitative thought experiment. Like this, we don't really, especially where we've upgraded to reduce nutrient loads during a year that didn't actually happen. It's also something though, when we think about the reason that these gates and the delta cross channel and the pumps were installed, it was to move that water across and prevent intrusion of more saline water. So it's a very reasonable thing to imagine. The magnitude is something that we didn't, at least our team wasn't quite aware of yet. So how will the regional sand upgrade influence that? It has the potential to substantially reduce those fluxes into that part of the system. I thought it was really intriguing to hear Keith's points today about the nitrogen fluxes and coming in, potentially those changes in fluxes, maybe that would result in, let's say, lower microsystem production. But it's sort of the fact that we're not actually simulating these events, I think makes it, we have to really be careful where we draw that line or where we draw that control volume. But I think it's a reason, it seems to me a reasonable quantitative way of approaching this issue. So the major take-homes, overall, the model performs well at reproducing spatial and seasonal variability in nitrate ammonium concentrations, both timing and magnitude. The model reproduces the Delta-Suisun, I didn't show this, but the model reproduces Delta-Suisun's generally low chlorophyll levels throughout much of the year. So we have growth, but we also have these dynamic grazers that essentially rise up and suck down that new production. We also have the strong turbidity throughout the system. And that we get this relatively low levels that we would expect 2 years with very different forcings and responses. And phytoplankton blooms, one of the things we don't get, and we've put a lot of effort towards trying to capture this, is there are some peaks that we really wanted to get, like peaks along at Decker Island and the Sacramento River, and we just couldn't quite get them. But that's where I think some of the sensitivity type analysis can come in. It's like, well, what would it take to produce a bloom there? It becomes an issue like very much related to some of the other Delta RMP priorities. It also does not capture the space-time windows of elevated phytoplankton biomass during 2016. We do actually do reasonably okay in 2011. So some of the limitations, well, we're not predicting biomass levels as well as we are nutrients. But something that I think is also important for us to think about is how important are those blips, the blips in concentration? Like we see high concentration, does that necessarily mean on a mass basis lots of production of biomass? Or is it a little puddle over here where you're getting phytoplankton to grow, but it's staying high because there's nothing grazing on it and there isn't a lot of flow through the system? So I think the shiny object of a high peak is something we have to be careful about. And it's also something that we can explore quantitatively. One of the ways we've been doing that is by calibrating the model using the flux stations and chlorophyll and nutrient levels from the USGS stations. And so trying to bring that together, we're also, instead of looking at just chlorophyll, we're comparing modeled and observed or calculated gross primary production. So trying to look at more integrated signals. No macrophytes. This is a bit of a catch-22 because, well, if the macrophytes are taking up a lot of nitrogen, and we're not simulating them, well, we shouldn't be trying to do better with calibrating the model because we're missing a key component. But getting those macrophytes in there is non-trivial, although I wonder if Zhenlin has a trick up her sleeve. But I'll stop there, and there's additional stuff I could cover. Some of the things that I think we're poised to be able to pursue is beginning to explore issues like, where are the places now? We're doing reasonably well with nutrient cycling. We're doing okay with phytoplankton production in certain parts of the system. One of the things, something that seems to be really interesting to explore is what are the different pockets in space and time where you can get a bloom to occur, or can we force the system to create a bloom? What does it take to actually produce that biomass from a healthy ecosystem supporting food webs? Something that I would hope we could pursue in the future.
We do have a few minutes for questions.
I didn't bring this up on last talk when we talked about it over lunch, but the cities of Turlock and Modesto removed their discharge to the San Joaquin River Delta. 25 and 80 combined. The city of Stockton's moving from nitrification, the full denitrification. So they're going to be removing nitrogen from their discharge, 30 mgb. What effect will that have? Those are, of course, the San Joaquin River and the transition.
Yeah, it's a great question. I think, well, if it just sort of took it linear, assume that it was proportional in the time period where DIN loads to that region, the time period when DIN, like so let's say summertime DIN loads to the central delta and the south delta transition zone, June through August, I think what was coming already from other parts of the system, not the central delta, were already negligible. And so it could be because, well, yeah, high concentrations in an area, but high concentrations that might not be accompanied by high flow. So not bringing that mass into the system. And so it's something that we'd have to definitely take a look at it. I wouldn't want to speculate, but I think given that it's already small, particularly here, I would say in this simulation, it would just be small for that region. For the interior delta, it would potentially have substantial effects in around the Stockton chip channel and other areas off to the eastern part of the delta.
There's a question on the audio.
It just said, if you have a question in the room, if you could stand up, it'll make it easier for the mic to pick it up. People they can't hear the question, or maybe the speaker can repeat the question.
Oh, sure. Yeah, the question was Turlock, Stockton, and what was the other? Modesto or
remove their load as of 2020.
Are all remove their loads as of 2020, and Stockton is moving towards denitrification in the coming years. And what would the effect be in those interior zones where aquatic vegetation and HABs are potentially problematic?
We do have a question online. So Joy, have your hand raised. Is that right? You go.
Okay, if you can hear me.
Yes.
Thank you. Dave, so I guess the story you were describing related to the SAC regional upgrades, and how big of a source is that relative for dissolved inorganic nitrogen relative to the other sources? I mean, does that really drive the algal blooms in your understanding of with your model? Is that a hypothesis that you think is better established now with the modeling than before?
So during I don't know that we would be able to say that it's driving the blooms, because there are a few of them in terms, let's say, major phytoplankton blooms or the cyano blooms that we weren't directly simulating. But I think if we were talking in terms of nitrogen loads moving along the Sacramento River during the summer months, it constitutes the vast majority of the nitrogen moving along the Sacramento River, dissolved inorganic nitrogen moving along the Sacramento River. So as that gets shunted off through the Georgiana Slough or Delta Cross Channel, then it's constituting a major source of nitrogen to the interior delta in these simulations.
Great. I'm gonna kind of cut us off there just to keep us on time. We will hopefully at the very end, if Sidra, you had a follow-up or anything, hopefully we'll have a few minutes at the end after all three presenters, so we can always circle back if there's more questions. So our next presenter is Joe Domagalski from USGS, who's going to be talking about the SPARROW model in his presentation.
You can go ahead to the next slide. All right, so I'm with US Geological Survey, and I'm going to be discussing our SPARROW model. And this is something that we've been having in concept to implementation over about a period of about 30 years now. I'm going to start out with a little bit of history about where this came from. So a little bit more than 30 years ago, the Congress of the United States asked the Environmental Protection Agency, how well is the Clean Water Act working? And EPA says, well, we really don't know. We don't really have a systematic way of understanding the effect of the Clean Water Act and status and trends and all that. So the Congress at the time says, okay, we're going to commission the US Geological Survey to do this for us. And we're willing to give them somewhere between $50 to $60 million a year which was nice money to do that. So we divided the country up into like about 30 major river basins at the time. We dropped some of them along the way. One of the first things we did at the start of the program was to bring together stakeholder groups that we called a liaison committee at the time. In fact, Tom Grofald might have gone to some of these meetings way back when. And so we would tell people, what we were going to do, what the program was about, that we were going to look at surface water, groundwater, aquatic biology. And then we would ask them, what are some of the major questions in your basin that you're concerned about? And just about every study unit in the country said that nutrients are our biggest issue. All right. Interestingly enough, the Sacramento River Basin said, "Oh, we don't care about that. We want to know about mercury. We want to know about aquatic toxicity." This is a long time ago. We didn't even have at the time, the national program wasn't even going to fund mercury, but fortunately, David Kravinoff was starting his career in Wisconsin. I talked to him. I convinced the national managers to give me money for mercury. And those were some of the first mercury and methylmercury measurements that we did in the Central Valley. All right, so going back to that liaison committee, they said, all right, so our big problem is we know nutrients are an issue, but it's really hard to figure out a way to implement targets. Most of these basins are unmonitored. So if we set targets, we need some kind of scientific basis to do it. And, if we're going to do different types of management actions and tell people to spend money, how can we justify that? We don't really have a way of doing that. So they asked us, is there any way that you could help us out with that? So we said, okay, we'll get back to you. We'll think about it. We commissioned our statisticians, hydrologic modelers, people that understood the biogeochemistry of the nutrients. And can we put together large watershed models that would tell us in unmonitored portions of a watershed based on a limited set of calibration sites, how nutrients move through the system from headwaters down to terminal bodies of water like lakes, rivers, and estuaries. Next slide. All right, so so what are some of the data gaps addressed by the study model? A lot of these questions from 30 years ago are very similar to what we've heard all day today. So that sort of thing hasn't changed. Go to the next slide. So as I said, the SPARROW model was developed to address data gaps. All right. It was obviously impossible to monitor all streams. The vast majority of streams in the United States, anywhere in the world, just have a small number of gauging stations and even fewer number of water quality monitoring stations. So we needed a model that was based on discharge, possible sources of nutrients, which we had somewhat of a handle on, and biogeochemical simulation to assess source, transport, and load. And we wanted to then use this model to understand factors that were affecting water quality up to that point in time, and then simulate water quality response to climate and land use changes. So the model that we wanted to do then would predict mean annual flux, yield, and concentration for unmonitored stream breaches and watersheds based on this limited number of calibration sites. All right, so next. So I think I'm the first one to show an equation. We'll go through each term in detail. No. So what we're trying to do in SPARROW is to measure the annual stream loads. That's kind of the key here. It's one of the limitations of the model. So we have SPARROW 1.0, which simulates annual stream load. We're currently working on SPARROW 2.0 that we call dynamic SPARROW. That's going to divide this up seasonally or monthly so that we can get better time steps. So we've seen a lot of talks today how things change seasonally. It's kind of hard to parse that out if all you have is an annual number. All right, so this is based on the sources of nutrients in a watershed. So things like atmospheric deposition, wastewater discharge, fertilizer, cows, so on and so forth, natural geology, something we call land to water transport. So from those sources, how much might go to a stream breach? Aquatic transport means like how much of this might decay along a stream breach. And then if nothing works, we throw in an error term, right? So, so the spatial reference is a stream network. I'll show some examples of that. And then the spatial element is something we call a stream breach and associated catchment. We want to be able to follow this down from like headwater streams, first order streams, all the way down to estuaries, and then develop an optimal set of rate coefficients that balance the material mass, source inputs, stream loads, and storage loss. And so also when we're dealing with a large watershed like the Sacramento River, that's heavily modified by human use, we have to also know the plumbing really well. So like we'll start off at the headwaters, somewhere along the way, there's going to be diversions. And if we're going to be able to calculate an accurate stream load along that reach, we have to know where the water is coming from, both naturally from precipitation, irrigation, and how it's being diverted. Otherwise, it's not much of a model. All right, so next slide. So this is kind of a conceptual diagram of what we're looking at. So we have these different sources like wet and dry deposition. So some of that information is fairly well known. The wet and dry deposition comes from atmospheric monitoring, along with how much rain falls on certain parts of the watershed. Urban wastewater, or all wastewater, it's fairly well known. That's all public record. We're looking at average amounts of wastewater discharge per year. And I don't think I mentioned it yet, but the SPARROW 1.0 models are designed to look at average conditions. So we've got stream monitoring at a set of locations. If we have a long-term record, at least 30 years, we'll look at that. How much is our annual amount of water flowing through our gauging stations? And then we do something called detrending. So if there's a trend in stream flow, we take that trend out to make it look like it's an average condition. That was what we decided for SPARROW 1.0. If there's our water quality at a gauging station, we'll look at the trends there and detrend that. So SPARROW 2.0 will change how we deal with that. All right, so we have other information like precip, temperature, soil permeability. Certain parts of the watershed, water is likely to infiltrate into the ground. If there's a lot of sand, you'll lose mass that way. If there's a lot of clay, you're going to erode clay during a storm and release nutrients. Geology is a source for some constituents like phosphorus. And then we try to get as good of information that we have on the farming practices. So nutrients applications on land, a little bit difficult sometimes to really get a good information on that. What we've done over the years is to take county level sales, look at the National Land Cover Database, say we've got a certain percentage of cropping land and certain percentage of the land in crops. We'll parse it out that way. It's not the best, but it's about the best that we were able to do. So we might be able to do the same thing with how many cows are in an area and so on. All right. And then these different terms like this geology, geohydrological factors will help us in our land to water transport, and the climate data, temperatures in the stream, depth of the stream, how long the water is in the stream will help with our downstream transport to reservoirs and estuaries. All right, so some of the pros and cons right up from the start. So we can help identify the options for reducing loads or nutrients. We can help design strategies for water quality protection and meet regulatory requirements. Predict changes in water quality that might result from management actions or other actions. So what happens if farmland is taken out of production, urban land grows? How is that going to affect how much nutrients are transported? We can predict sources of nutrients in specific locations based on a detailed hydrological network. And the model takes into account natural and human altered hydrology. So the cons are data is intensive and you must have accurate information. If all of your sampling sites were along the mainstream of the river, but you had a complex land use upstream, the model's not going to work because you're not going to know how much nutrient might be coming from a farm, from an urban location, from a first order stream. As I mentioned, SPARROW 1.0, we detrended conditions, we detrended loads based on average. And we didn't have seasonal variability. And then also our models are only based for total nitrogen, total phosphorus, and sediment. So we can't simulate ammonia, nitrate, and that sort of thing. So next slide. All right, so I think I've already gone through this. I don't want to mention this again. So some of the data layers that are used, like wastewater, atmospheric deposition, and so on, and geohydrological factors and climate. So next slide. All right, so our stream network that we use now is the National Hydrography Data Set. All right, so when we first started, we had the EPA River Reach file. That was okay, but a lot of work went into the NHD data set. A lot of money went into this. This is the best mapped, best set of mapped watersheds in the country. We got exact information on location. If you look closer, you can see that there's first order streams all the way to stream outputs. NHD provides information on the exact location and the direction of flow of these watersheds within a larger. Next slide. And then we look at this a little bit closer, and this might be a little bit hard to see in the back. But if you look closer at these watersheds, this is for the Tahoe area. Each of these sections of a stream along the flow path is housed within a polygon that we call a catchment. All right, so this becomes our frame of reference for moving the water. So for our 2002 model, we had 142,000 of these catchments throughout the watershed. And we had 89 calibration sites. So that's what I mean about how much of the watershed can be modeled based on calibration. And so for each of those sub-catchments in a bigger watershed, that goes into our data frame for our model. So in column A, each of those watersheds has a number. And then each one is going to have some type of information. If there's a wastewater treatment plant, how many kilograms in a year do they discharge? What's the average temperature? What's the average stream flow? What's the rate of flow? What's the average depth of the water? So on and so forth. If there's a diversion along the flow line, we have a column there, diversion, yes, no. If yes, how much? All right. If it's a calibration site, like where those red dots are, then we say calibration site, yes, annual load, so many kilograms. So that's how we start with the model. Next slide. All right, so as I said, calibration sites must be co-located with the stream gauging stations. We need a sufficient number of samples collected over the year to calculate an annual load. So 1 or 2 samples is not going to cut it. We usually use another model like LOTUS that some of you might be familiar with to calculate an annual load. If we don't get a good statistic on that, and we just can't really use that location as a calibration site. So we had 89 locations for our initial model. And then these calibration sites have to be located near important land uses or hydrologic regions in order to build a statistically significant relationship between stream load, land use, the land to water transfer, and aquatic decay. All right, so these were our model domains for 2002 and 2012. So in 2002, myself and one of my colleagues put this model through. for total nitrogen and total phosphorus. And then in 2012, we handed that off to one of our colleagues in Portland, Dan Weiss. And what they decided to do up there was to take out some of the areas that we had in 2002, like the desert, because there's no water there anyway, so who cares about it? And limit those areas, and then include up through the Willamette Valley and up through areas throughout the state of Washington. So on the left, the 2002 models, which where our calibration sites were. You can see where some of the major wastewater treatment plants are. If you look closely, where the regional sand is, and you can see all that green around San Francisco Bay. They don't really need a SPARROW model. They know where their bad stuff is coming from. All right, so next slide. And then water diversions. So we had a pretty good set of knowledge of water diversions in 2002. We drilled deeper into the system to see what was going on with water diversion. So in 2012, Dan included 91 for power supply, 248 for irrigation, 642 for municipal water supply, and 72 for other in-stream transfers. So we really kind of did our homework there and came up with as good of a representation where all of these different diversions were located. Next slide. All right, so I guess here's mine that you can't read. So model evaluation, statistical summary. So when the modeler runs this, they have all those different possibilities of sources, land to water transfer, aquatic decay, and then the model spits out a p-value for each of those. So a p-value about 0.1 for source, land to water delivery, or aquatic decay term is considered insignificant and is taken out of subsequent model runs. So this was from our 2002 models. We also did an R-square of the overall load, which in this case was 0.994, pretty good, and R-square for the yield, which was 0.84. And so the significant sources that we had back in that model was atmospheric deposition, fertilizer applied to farmland, manure, wastewater treatment, and one or two others. In land and water decay, precip obviously was an important term. About a percent sand was significant here. It's hard to see, but for a percent sand, there's a negative coefficient. So negative coefficient indicates a loss. So a lot of nitrogen, as we know, is lost from the Central Valley, from the water, and it ends up in the groundwater of San Joaquin Valley, right? So that's why that's a negative term. And then streams of different sizes then were also significant for aquatic decay. And then we looked at other statistics as well. We had something called a variance inflation factor. So that came up high. That indicated that one of these terms then was causing collinearity, and we took it out of the model. So the modeler has to go through like quite a few iterations of this to optimize which are the best sources and what are the best terms for land to water movement and aquatic decay. So next slide. All right, so some of the things then that we can do with this is to develop heat maps of these of the sources and how this moves through the area. So for each of those catchments that are identified, we first have an amount that we think is being applied right at the catchment and how much is delivered outside the catchment. So the amount that is potentially supplied within the catchment then undergoes those biogeochemical equations where we lose some of it. And then that amount is then moved down to the next catchment that picks it up and it moves it further downstream, undergoing further decay if that is the case. So on the left is total nitrogen from Freeport and total phosphorus at Freeport. All right, and the bar chart shows the main sources. So for so light blue is atmospheric deposition, green is fertilizer, and the red is the wastewater treatment. All right, so in the San Joaquin Valley, the green bar, not surprisingly, is the biggest. And then in the San Joaquin, in the Sacramento, wastewater is a bit more important than what we see coming out of the San Joaquin side of things. All right, so then on those heat maps, we can see that the colors start to darken within the Central Valley. Not surprisingly, up in the headwaters, they're much lighter. All right, next slide. All right, then we can look at some other locations here. So that blue arrow is for the lower Sacramento. This is from our 2012 model, which included the entire West Coast. And so we can, so that blue arrow gives us the same bar for the Sacramento River at Freeport as what we saw in the previous slide. And we can compare it to some other coastal locations. So the highest yield that was modeled in with the 2012 model was for the San Francisco Bay. Second highest, Ventura, San Gabriel. Third highest, Santa Ana. And the fourth highest, Laguna, San Diego. So those areas very much dominated by wastewater discharge. All right, so next slide. All right, so here's something else that we developed for the use of this model. I call it a canoe trip down the river. And how we did this was we isolated from the model those catchments that were right along the major rivers. So for this case, the Sacramento River from the headwaters down to the estuary. Same thing with San Joaquin. So looking at the San Joaquin, for example, starts out with atmospheric deposition. Then we head into the valley, and that straight line up is from fertilizer. Then that downward part there is from a water diversion. I forgot where that water diversion happens. And then things start to build up again in the San Joaquin Valley until we get to the Delta. And then the one on the far left is for the Sacramento River at Freeport. It starts out with most of the sources coming from atmospheric deposition. We enter the valley, it jumps up with farm fertilizer. And then wastewater treatment loads are fairly low throughout the Sacramento River until we get to regional sand. And this was before the upgrades to the plant. So that's why that red line then shoots up for the Sacramento River. And then we can see a similar type of thing for phosphorus. So some other people throughout the country did other things with their models. For example, some parts of the country have a number of reservoirs. Some of them are eutrophic. They had these annual loads of phosphorus. They use something called a Vollenweider model, which some of you might have heard of, which relates total phosphorus coming into different reservoir characteristics that would predict chlorophyll A, and that they thought that might give them a management option for how much phosphorus they would have to remove from the watershed to bring the chlorophyll A levels down. That's just one other option that they used. So next slide. All right, so how can future research projects inform data gaps? Go ahead to one more. So this is my last slide. So these original SPARROW models invoked annual loads with detrended discharge and climate data. So we're working on what we call a dynamic model right now. Our national team is still together. The National Water Quality Assessment Program has sunsetted, but we kept the SPARROW team alive, just put them in a different management box. And so they're working on what we call a dynamic model. This is going to be not a model based on a particular base year, but a multi-year model based on how many years of calibration you have. So the national team is working on the Illinois basin as their test site. So it's a river that flows into Lake Michigan. And they have like around 30 to 40 years of water quality. And they're trying to see how they can optimize that model based on those 30 to 40 year scenarios. So I'm working on another basin up in Oregon, Upper Klamath. We have about 20 years of data on that, well, actually more than 20 years on that one. And water's moving down to Upper Klamath Lake. And then as a worst case scenario, I'm also working on Clear Lake, which has probably the worst amount of water quality, and whether or not, I mean, bad water quality in the lake, but not very much water quality in the watershed. Can we actually use this? We got a plan B if this doesn't work, but we're going from testing it out in basins with a lot of good data to basins with hardly any data, and what can we do with it? So I think that's it.
Thank you, Joe.
Joe, when you talked about putting fertilizer, you talked about fertilizer being the biggest source in the Sac Valley and Salaquis Valley. But on fertilizer, there's lots of places where fertilizer can go.
Right.
Down, can run off. You know, denitrification can go off, goes off at harvest. Is there an assumption as to the amount of the original, because you're just using fertilizer sales? And this would occur for urban, dairy, irrigated ag. Is there an assumption about there's some percentage of the actual application amount that's then assumed to be able to run off? Right.
So there's amount that we assume that might get sourced within a catchment, but then we apply those biogeochemical terms and geohydrologic terms to assess how much is going to run off. So in areas with a lot of sand, for example, we see a lot of that nitrogen actually goes into the subsurface. So that's why we got, and it makes sense because we got so much of the San Joaquin Valley in sandy soil areas with a lot of nitrogen fertilizer in the soil. So areas with a lot of clay soils indicate that a lot of that phosphorus that's applied will move off the land. So yeah, that was the idea there, how that is to be handled. So I'm probably out of time, right?
Yeah, I think so. I don't think I have. Do you have a quick question?
A quick one, yeah.
Okay.
I think Debbie asked the question before about atmospheric deposition, and we mentioned it a few times. Is there a rule of thumb when, for a certain amount of loads to a system, maybe aerial loads from water inputs that you do or don't need to consider?
Well, it's whether or not it turns out significant in the model. When we tried running this for 2002, we couldn't get atmospheric deposition to be so register significant in the model, but forested land did. All right, but then when we compare like where these areas are, they're pretty much the same. So it's kind of like how the calibration works out. Yeah, good question.
Thank you, Jen. All right, so our last talk of this session before we take a break is Zhang from DWR, and she'll be speaking about modeling delta water quality using coupled hydrodynamic and biogeochemical models.
Thank you. Hello, everyone. My name is Zhang. I work for the Delta modeling section at DWR. Today, I'm going to present modeling delta water quality using coupled hydrodynamic and biogeochemical models. Thank you. Thank you. So first, I would like to acknowledge the contributions from the collaborators who have the project has really benefited from their contribution in ideas, discussions, and data they provided to us. So on this slide, I'll just start with like an overview of the water quality issue. Actually, I think the presenters this morning has already gave us such a great overview. I'm just really repeating what they're saying, so please bear with me for a second. So I'll just talk about the water quality issue and how I think the mechanistic modeling approach would be a great fit to tackle those water quality issues. So our delta system is a high nutrient environment, and the presence of the nutrients has the potential to fuel the growth of the primary producers. Right, so which includes submerged aquatic vegetation and phytoplankton. Also like benthic algae and other types of vegetation that I haven't listed here. So having a phytoplankton bloom event, particularly like a diatom bloom, is actually great for the fish. However, as the phytoplankton die and the breakdown of the tissue can consume oxygen in the water column, that can cause low dissolved oxygen after an excess bloom event, which can result in things like fish kill. And Another thing is the cyanobacteria we talked about this morning, which is the main concerning type of harmful algae in the system. And the harmful algae can produce toxin, which can also result in mortality of the fish and other kinds of health-related issues. Yes, thank you. So the environmental factors being identified to influence primary producers are light, water movement, and water temperature. And one thing I'd like to mention is that primary producer can also in turn affect those environmental factors. For instance, the presence of submerged aquatic vegetation can affect water movement and the light availability through shading. So in addition to nutrients light, primary producers can also be affected by grazers. And in our system, two types of grazers are important. They are clams and the zooplankton. And the presence, so both type of grazers will consume phytoplankton for food, and the presence of clams compete with zooplankton. And because the presence of clams can reduce the amount of zooplankton available and therefore less fish food. So that can result in less fish biomass in the system as well. And historically, this has actually been observed that the introduction of some of the invasive clams has coincided with a sudden decline in phytoplankton and fish biomass. And so next, I would just like to continue to finish the biogeochemical cycle is that the mortality of primary producers produce detritus, and detritus through remineralization returns nutrients back into the nutrient pool. And detritus also interact with grazers. Mortality of grazers can produce detritus, and some grazers can also graze on detritus, like clams can have detritus as a food source. So, so this is like a lot of things going on this slide, and I would like to emphasize that next. That the environmental factors, the biogeochemical process, and the water quality issues, they're all interconnected. So I think as our previous presenter mentioned that we need a holistic and a systematic approach to tackle these water quality issues. And one such approach is a mechanistic model. And so tackling everything here is a really big list, it's very ambitious, it's our goal. But for now, we're gonna start with just the core process, the most important processes. And these are the 4 components listed on the slide here, the nutrients, primary producer, grazers, detritus. And by the way, these are also the 4 components are also the major core model for more complex biogeochemical models in an aquatic system. Thank you so much. This is a lot of okay. So, so now we have a biogeochemical model, but that is not enough to resolve our water quality issue. We also need a hydrodynamic model. Please. And this is because we need to be able to resolve also the movement of water. Just because we observe a water quality issue happening at one location, for instance, in French Tract, it doesn't mean it happened locally. It can be originally somewhere else and transported to the observational site. So we need to be able to resolve the process of transport, mixing, and tidal dispersion. And that's why we are applying a coupled hydrodynamic and biogeochemical approach. So one thing I'd like to mention when I mention coupling is only just one-way coupling. That means the output from hydrodynamic model is used as input for the biogeochemical model. However, the specific hydrodynamic model platform also allow two-way coupling, and that means things like submerged aquatic vegetation is affecting the flow, and that impact can be integrated through a two-way coupling approach. So next, please. So the specific modeling platform we're using is called Schism cosine. So Schism represents semi-implicit cross-scale hydroscience integrated system model. And cosine stands for carbon, silicate, nitrogen ecosystem. So here's a list of reasons we chose this particular modeling platform. And first is open source. It's unstructured grid model, so it's very well suited for the irregular geometry in our system. It's a very well supported model, community-based, and by Virginia Institute of Marine Science. And second is a very important reason that the model has been very extensively calibrated for the Delta system for the last 10 years by our Delta modeling section. So the model has already been used in a lot of applied studies. For instance, the Delta Conveyance Project, the Emergency Drought Barrier Project, and incident take permit for the State Water Project. So these are just some examples. And moreover, we are also in the process of developing an operational model. So that means we'll be able to model the flow in near real time. So that's a great feature we're working towards. Third thing that this particular modeling platform has already been applied in the Bay Delta system, and at least 3 peer-reviewed journal articles being published. And lastly, it is coupled to DABM. It is a framework for aquatic biogeochemical model. It includes 20 different biogeochemical models. So that means that we can swap in and out different models that suits the need for our particular study interest. So next, please. So the biogeochemical model we're using is a very simple model. It's called cosine. It has 13 variables. So in my opinion, it's really just like a minimum list of biogeochemical model of variables you need to model the system. It is super nice to use, and you can extend it. So it includes the 4 components I mentioned on my first slide. So these are nutrients, phytoplankton, zooplankton, detritus. So nutrients, we have nitrate, silicate, ammonia, and phytoplankton. We have 2 groups, small phytoplankton and diatom. The difference between the 2 is only that the diatom consumes silicate and the other one doesn't. And for zooplankton, the microzooplankton grazes on small phytoplankton, and mesozooplankton grazes on diatom. And then the detritus, we also model phosphate, dissolved oxygen. We care less about the last 2 variables. In addition to that, we also have a clamp grazing module. In the way the clamps are not modeled dynamically, so we use it as a model input. We specify what's the grazing rate at the bottom. We also have like a simple sediment diagenesis model that models the return flux to the water column. So calibrating such a coupled hydrodynamic and biogeochemical model requires extensive data set. And in my opinion, this is the most exciting and a collaborative aspect of this project. So in the next 2 slides, I will go over all the data that we have used to run the model and calibrate it. I'll start from the hydrodynamics. So this is our model grid. And these are our model boundaries. On the ocean side, we used observational data of water level from 2 NOAA stations. And in addition, we also used the model output from a regional ocean model. Use the model output of salinity and temperature to drive our model at the boundary. And for the delta side, we used a continuous observation of water level, flow, temperature, and salinity. Next. We also implemented detailed gate operations. So these are all the gates that's happening in the delta system. And then these are also the delta channel depletion model. So the delta channel depletion model represents exchange between the island and the channels. So these are important for things like agricultural irrigation, the return flow, and the seepage. So in addition to in addition to the data, we also compile a rich data set for wind. These are from 79 field stations across the system. And we also have a data set of everything that we need to calculate heat flux, which includes air temperature, solar radiation, long-wave radiation, relative humidity, and air pressure. So that is a lot of data. So, okay. So next I'll go over all the biogeochemical, the data we compiled to run the biogeochemical model. So first is the BOTW. So this represents discharge through the wastewater treatment plants. We have around 50 to 60 of those stations, and we have developed a pipeline to grab data from eSmart, which is the data portal from the Water Board. I've also been communicating personally with Tim for the regional Senn data. Thank you so much for sharing with us the data, which is fantastic. And so next is the discrete sites, discrete sampling data. So this data set is really important. So mainly we're using that to validate and calibrate, to provide boundary conditions and validate our model. So the data sets come from EMP and IEP, so 2 different sources. And we're using mainly the nutrients, chlorophyll, and DO data. And then we're using the continuous turbidity sites. They're continuous turbidity data as a proxy for the light availability for the system. And so these are from USGS and DWRDES. And then we have clam grazing data from 3 different programs. I'll give you more detail in the following slide. Next. And then lastly is the zooplankton data. And so far we haven't used zooplankton for the model validation, but I really appreciate special credit for Rosie Hartman, who has put together and led this effort to compile a very rich data set from different sampling effort for zooplankton and convert it to zooplankton biomass that we can actually use to validate our model. So that's great. Next, please. So on this slide, I just give you an introduction on the light field we're using the models. So the light field, so light extinction coefficient controls how much light is available for phytoplankton growth. On the left is what I'm showing you is a calibration between the observed turbidity in FNU and TNU unit, and the y-axis the light extinction coefficient. So this data set is from EMP, and basically you can see that the 2 variables are highly correlated, and with the coefficient, light extinction coefficient is about 1/10 of the turbidity. So we're using that relationship, and also on the right is the continuous observation of turbidity throughout the delta system. And then we interpolate spatially to create this continuous map of light field. And the spatial interpolation method we use is called FDAPDE. That's a special method that takes into account of the irregular geometry. So it's very suited for the system and has been applied to quantify delta smelt habitat in the past for our environmental permit for the state water project before. Next, please. So the clam grazing data, as I mentioned earlier, comes from 3 different programs, and the acronyms for each of them is at the bottom of the slide. So this is really a fantastic data set. However, we have very good coverage in the center of the delta and the Suisun Bay. We do have some data gap, and one area we do not have much data is in the Sacramento region, even though I learned today from Tim and Lisa that actually they have done, yeah, which is great. Hopefully I can get your data. Yeah, that would be fantastic. And then we also didn't have much coverage in the South Delta region. And another thing that's unfortunately this data set stops in 2019, which means afterwards we will have to make some assumptions for now. So there are 2 types of clams in the system. That is corbicular, the other one is potamocobular, and so we just did a spatial interpolation basically using the observational data. Yeah, again, I'd just like to emphasize that this is really based on a very simple inverse distance weighting method. So there's a lot of uncertainty in the area that we do not have any data, even though the field is continuous. Next. Yeah, so before I show you my validation or model result, I just want to say that this modeling effort is built on previous modeling effort. So this is our modeling domain, and the previous modeling effort is a study that we collaborated with Virginia Institute of Marine Science, and their modeling domain is next, please. So that is their modeling domain. So you can see that they have a more focus on San Francisco Bay. So the model biogeochemical coefficient was literally taken from that study, and we haven't done any tuning. We just take whatever they have and run the model for the delta. Another related study happened in Cashew. Next. So that is the region, and I think Dylan this morning talked a little bit about the project. So that project has a focus on the submerged aquatic vegetation, and so I would like to talk a little bit more about that because currently for our schism model, we have considered the impact of submerged aquatic vegetation on the flow, mainly in the front tract and the cash flow region, but we haven't modeled the dynamics, like the growth and mortality of SCV in a dynamic way. And so although that is one of our focus in the near future is to be able to extend the study that happened in cash flow to the entire delta. Next, please. So these are our modeling results. The dots represents the discrete sampling data and the lines represent our model. And we have a lot of stations, but generally the left on the left of the screen is the north of the delta and the right of the screen is the south of the delta. So I think our model generally produced the seasonal cycle that you observe from the discrete data. However, some of the south delta station, our model predicted neutral limitation. That doesn't happen. And one reason is that I think I mentioned earlier on that the South Delta is a region where the clam grazing rate is very uncertain. We just have very little knowledge. Another reason is that that region has a lot of water diversion and return flow, and that impact hasn't been well quantified in the model yet. So next, please. So that is the result from ammonia. Next, please. And then that's the chlorophyll result. So I would like to take a little bit slower here. So the two left column is north central site, and the two right column is the south delta site. So I would like to point, probably you can't see the stations, but I would just point that the C10A, which is the one on the top, so that is our model boundary from the San Joaquin River, and the chlorophyll A went up to over 100 microgram per liter. And I think one of the earlier speakers also pointed out that's something that happens pretty frequently, that not every year, but we can expect that sometimes we would have like a summer bloom event at the San Joaquin River boundary. And as the chlorophyll comes in, it gets dispersed, and some of the phytoplankton died and caused low DO issue in that particular region. And I think our model generally produced that the magnitude as the phytoplankton goes through the system in the South Delta. And in North Central Delta, I think this is the model results, probably you can see is for 2015. So generally it's a non-bloom event. I think our model produced that most of the time. However, there are two stations we're overproducing, which is the Steamboat Slough, which is the second one on the first column, and the Isoton. The reason is that these are the two stations still quite close to our model boundary, and there's still a lot of uncertainty associated with boundary condition, and we are still tuning the model, and hopefully we can definitely improve that in our future calibration. So next, please. And that's phosphate. Yeah. And next, please. What to say? So phosphate doesn't change very much, and then the DOs again doesn't have too much of a variation. Please. So conclusion. So so far, the model development has been only going on for 1 year for this particular project, and this is very much an infrastructure level of modeling effort. We're building the capacity to do the work, and we're not trying to calibrate for a particular year or trying to do it for a particular project. We just did a very simple test run and with the out-of-box coefficient that hasn't been tuned. And so the test year is for 2015. And I think it shows that the schism and cosine is capable of modeling the seasonal variability of observed nutrients, chlorophyll, and DO for most part of the domain. And definitely further tuning is required to improve the model. So the future work, definitely we would like to be able to calibrate the model more to make sure that it works. Particularly 1 year that we're really interested in is 2021, and I will explain a little bit more on that. We also want to apply the model for long-term calibration. We have 10 years of well-calibrated hydrodynamic sitting in-house. That's from 2008 and 2018. We want to be able to see that if we can use this coupled modeling approach to produce the 2016 and 2018 bloom event observing the system. Next, I would definitely like to explore that if we can validate the zooplankton model, we can validate the model with the zooplankton data. I also haven't used the USGS, the high resolution mapping data, which is a great data source that we would definitely like to use in the future. One solution to tackle this uncertainty or data gap for the clam growth data, the clam grazing data, is we may develop a dynamic clam grazing model in the future, and that I think that feature is already in the model by SFEI model. And lastly, one thing about HAPS modeling is we can't model HAPS at the moment, but HAPS is becoming a focus at DWR. And it's likely to be a focus in the next few years. So we're still thinking about if we should use more a deterministic, mechanistic, or probabilistic approach. And that's something I would really like to have a discussion with the HAPS expert like Kate and Alan. I think really appreciate your presentation as well. I learned a lot. Thank you. So next slide. Just want to explain why 2021 is going to be our next target year. And first of all, there is a confluence bloom event observed on April 23rd. So this is a data set. Did I say something wrong here? No. Produced by, San Francisco State University, observed by Dick Dugdale and Francis Wilkinson, and they observed that the bloom event happening in the confluence. And I think I talked to Brian before, and he mentioned to me that confluence bloom event actually is something that happens quite frequently, not every year, but it's definitely a phenomenon that we observe. And next thing is actually there was a diatom bloom event in May 2021. And at the same time, there was nutrient limitation observed. So there's a better map on the right that's from USGS Delta survey. And so it shows that there's a high magnitude of chlorophyll and coincide with a nutrient limitation condition in that region. And in addition to that, there are a few things happening in this year: the upgraded regional Senn, the emergency drought area on Falls River, and there's also the HAP event observed in Frank's Trap and beyond. I think there's also HAPs are observed everywhere in the interior of the Delta. Okay, so the last thing is about the data gap. Yes, so rather than call it like a data gap identified or inferred from the study, it's more like a wish list that I wish I could have better data sets on those items. First is definitely the clam grazing data, and special credit for Betsy Wells and Jenny Thompson. They have really done great work in leading the effort to collect this very valuable data set for the system. And I just wish that data could continue in the future. And the next one is the data on light attenuation. I definitely realized that the calibration I'm showing you, the relationship was based on the data set of EMP, and it hasn't been calibrated for each continuous sensor. I don't know if there's any error range associated with that particular relationship that can be extrapolated and cause the model to not work properly. Next one is continuous chlorophyll data. I think I talked a little bit during the break with Brian. And so this data set is collected both by USGS and DWR-DES. And I would really like to use this data set to calibrate the model. I think there are sometimes shows some discrepancy between the continuous data and discrete data. And I'd like to have a further discussion with Brian and Tamar and also the folks from DWR-DES to see if we can somehow resolve some of these discrepancies and improve possibly the calibration of the data. So I think that's the last page. Thank you. If you have any questions, thank you.
Any questions in the room?
Oh, I see a hand up.
Okay. Let me do one in the room and then we'll go to one online. Go ahead, Dave.
Zhenlin, thanks. That was great. I was curious what your opinion was going to be on clam grazing and a dynamic model, or of course, I feel like that's been a thorn in our side for a long time, trying to get that right. And now that you've had the chance to start over and do it with as an external forcing, what's your thinking on the best way to do that?
Well, I think if there is data, I think it's great to use data. But when there's not data, when we don't have data, definitely we will need to find a way to extrapolate, and using something cheesy like inverse distance weighting is not the best way to go. But if the model dynamically using a model based on food availability, at least that's one thing. I think we have better chance of getting the process right. So I'm very much in favor, even for my current model, to build a dynamic climate grazing model, maybe a simplified one, not as complicated as the DEB model, to be able to capture that process. I'm very much pushing towards that, and because it's not just for the model, the hindcuts, but also for scenario runs, like maybe historical runs, or what like when we want to play what if games in the future, we would need to be able to model the process dynamically, since it's such an important component. So
Let's do one on one and I'll come back to Ted.
To Joy.
Oh, thank you. Thank you, Zhenlin, for a great presentation. One question I had, do you folks at DWR and SFEI have a plan to compare notes? Like I know the modeling period that you showed looked like it was calendar year 2015, not water year, but somewhat overlapping with the SFEI model run. So is there an opportunity to compare notes to look at, like processes, do similar kind of control volumes to identify, understanding of loadings? That was one sort of a high level question. I think it's a great opportunity. So, and I know you've worked with both groups, so it would be a natural fit for you. But in addition, I was compelled to ask, what about macrophytes? I didn't hear you mention that, and I know that's a big gap there particularly in the South Delta. So just those two open-ended questions for you. Thanks. Great presentation.
thank you. So first question about the nutrient load. I think yes, definitely if that's required by a project, and if we want to learn more things from the nutrient transformation and how it's transporting to the system, definitely that would be something. We'll have to adopt a very similar approach as what Dave just presented. And so I agree with that. Second, did you say macrophytes? Did I hear that correctly?
Yeah, macrophytes.
Yes. So at the moment, we've only have this, well, not for this particular model. So this is cosine. There's another biogeochemical model that's already coupled with schism called ICM. And that model includes a submerged aquatic vegetation module in it. And I think I mentioned in the cashew complex, that project, Schism and ICM was able to resolve the interaction between like nutrient dynamics, for instance, nutrient uptake by submerged aquatic vegetation, the growth and the light effect because of the shading effect because of macrophytes, and as well as the feedback impact on the movement of water. So this process has already been achieved by the modeling platform, not by our current model. So I think, as far as I know, SAV is the only category of macrophytes that we've considered at the moment, if that answers your question.
Yes, thank you.
So I think we have how much, we have 10 minutes at the end, right?
Oh, right, till 3:10.
Yeah. So I know, Ted, you had a question for Zhenlin?
Oh, I was, when you're talking about the clam, it was the clam, the things that you had a list of things that you were hoping for. How would the monitoring programs go about collecting some of that data that would allow you to calculate that? Is there, I know Betsy's talked about doing some additional measurements that would allow you to constrain that better. Is there anything else that the benthic monitoring program? I think EMD is one of the only ones that consistently does it. So
Yeah.
That you would suggest that additional data that we could collect that would help you do that.
Yeah, I think at the moment, the best data set is the plant biomass data. And I think the clam grazing rate, I think my memory is calculated based on the clam biomass with some sort of conversion between the two, right? Yeah, so I think clam biomass is what's the most useful for us. And I know that's the most label intensive type of data as well. Yeah, so I remember last time I had a conversation with Betsy, she mentioned that there is some like survey data or remote sensing technology she's talking about developing, like so that we can do like survey in a bigger region. I think she said it's not going to happen in the next year or something, but I definitely hope that that would be a great development. So if that can happen.
So we can open up questions to any of the speakers if anybody has additional questions. Tomorrow, you started, so I haven't started to.
Just for the modelers in general,
Oh, yes, and project.
for the modelers in general, just commenting on how the modeling platforms are going to handle the extensive wetland restoration projects that are coming online that will also, impact both the hydrodynamics and the biogeochemistry pretty significantly. And yeah, what your thoughts are on that as we look to the future.
Okay, so probably I'm trying to answer for other people working in the group. So I participated in this project that we created, like we developed the grid, not just for the channel, but also for each individual islands in the Delta. And that the purpose of the project initially was because if there is like a levee breakdown and then island can be flooded. And I was told that I need to come into office at 2 a.m. if that happens to do a simulation. Be prepared, that happens. Yeah, so we are working towards that. Project is not done yet. So we're working towards building the grid for not just the water at the moment, but also the larger part of the Delta. So in my opinion, like the wetland restoration could be that we just need to extend our model grid to include those regions. And then it depends on if it's going to be like a scenario model effort or something that's actually happened. But for both, we already cover the region in our models. We'll be able to model the flow impact and biogeochemical processes as well. So that at least I think model capacity is there, but whether we're going to be able to calibrate a model is a separate issue. So we probably need a lot more data. For instance, sediment diagenesis process might be super important for those regions. And obviously I haven't included any of that in my model yet. And definitely I want to talk to you after the meeting to possibly include your observations and to refine our model. So.
Before we get to Debbie and Janice, I saw both of your hands jump up about the same time.
Oh, brother. Okay. So this is my question to the modelers is there seems to be some sort of overlap, but not all, there's certain purposes. If you would maybe describe how those different models can either work together or be used together to, to really come up with as much as, the best science that we can get in the near future as we move forward. It's kind of a hard question. But.
Well, one of the things I mentioned community model, but I was modeling, put an ing at the end because not being someone who comes from the modeling world myself, something I've learned a lot over the last several years is that everybody has their favorite model, and you're not going to necessarily get people to switch over to something that you've developed. So that ing at the end is important because it means that there could be other ways of collaborating with anything, like all the data inputs that Zhenlin mentioned, like those are incredibly challenging to pull together and assemble, and like so there are ways of using different engines, but the same fuel. And so I think that's one of the ways I think it especially with groups that have different missions or different needs, like TWR, especially if they're going to go for more of an operational model for the forecasting of flows and flow routing that they then use for the biogeochemical model. That's something that another program that's interested in nutrient cycling can never quite keep up with all the information demands. And So I think there's using open source. So I think there is a possibility of moving in that direction, like of actually sharing those models themselves. That's
Madison and Terry?
My question is for the model. And it's great because you've got all the details. What do you have to do to update it for funds?
Well, each of the questions that I mentioned, has an average annual temperature. So if you, so you can project into the future, what these temperatures might be. All right, so, we're actually doing that right now in our Clear Lake study. We're not using SPARROW to do it, but we're, one of our collaborators on that, Michelle Stern, is, working on, the next 100 years of the Clear Lake Basin, for example. She'll be able to tell us how those stream temperatures will change over this period. And so we can apply that anywhere really. And so that will change the, the biogeochemical reactions. It will change the number of parameters as the nutrients move downstream.
And precipitation pattern.
Oh, yeah, right, all that.
Terry and Malcolm, you.
This is just a great discussion and also the collaboration, which I think is important. On some of the discussions, obviously some of the modeling is going to be looking at echo water with regional sand when our treatment plant upgrade comes into place. But I also wanted to just let folks know, some of you may or may not be aware, we also have another program called Harvest Water, which is a very large water recycling program that will be taking 50,000 acre feet per year and then delivering it to agricultural lands in southern Sacramento County. And it also has some environmental ecosystem benefits and water quality benefits. So during that summertime, a majority of our water will not be going into the river during the summer season. There's also wintertime use of that recycled water, but it is a large volume of water that will not be going in, to that. So as you're looking at different loadings. So just wanted to share that. So keeping that on folks' radar as you're doing the
When is that coming online?
We are in design now and construction is anticipated to start in 23 and hopefully delivering recycled water in 2025. So we received a water storage investment program grant of about $291 million. So that's moving forward. So we're real excited with the program, but just wanted to throw that out there. And as you guys are doing things, I'll be happy to answer questions. Thanks. Brian?
Hi, once you hear virtual question, if folks can hear me.
Yes.
Yeah, still struggling with the hand wavy thing, but great talks. I really enjoyed this session. A couple of questions, both related to timescales and processes associated with that. I'll ask the first question pointedly to Joe because their SPARROW 2.0 with the dynamic SPARROW model that he was talking about sounds really interesting. And then once we hear from Joe, I'd like to ask the second question because I don't want to, confuse folks with two broadly thematically same but very different questions. So first question for Joe. You had a very interesting slide where you talked about this canoeing along the river sort of concept. And when you're talking about that in a sort of annual averaged model sense, then the fluxes that you have to look at are essentially external fluxes that are coming into the system and perhaps advection and diffusion or some sort of mechanistic transport along the multiple linked watersheds. Now, when you have seasonality built in, you might also get things like intermediate term holding and then reintroduction of mass into the system over time. So do you have mechanisms to account for those types of fluxes as well when you try to do this sort of analysis, this along the river sort of canoe analysis in SPARROW 2.0? And if so, would you be able to share some of the strategies you're thinking about?
Yeah, actually, that was pretty good insight because one of the things that we're trying to do in 2.0 is to have what we call a storage term. So we heard something earlier today about legacy phosphorus. it would be kind of like a similar situation like that. So over a number of years, we would have some idea as to how much phosphorus was input into the system over the years and how much came out along the way. And then there would be a certain amount stored either in sediments or in stream channels. So then if we go into subsequent years, that storage term, then we might see that in our canoe trip all of a sudden being like a big flux after a big precipitation event. So yeah, the answer to your question is that's going to be part of it, and it's what we're calling storage, either storage or a lag for when the phosphorus then or nitrogen would re-enter the system.
Okay, thank you. Yeah, that should be pretty cool to look at. So the next question that I had was for David and for my apologies, David and Zhenlin. Zhenlin. Yeah. So when you're modeling the system in 3D, obviously with close to 1 million cells or more, when you have things like wetland restoration, somebody brought that up. You also have long-term water diversion operations planned, climate change, sea level rise. There are bound to be impacts to the mass sequestration and release in time scales that are much longer than simulation time scales that are typically possible with a model with close to 1 million cells. So there's obviously going to have to be some type of parameterization of those long time scale processes. So do you have any insights into how you plan to tackle those problems, or are these models just not the appropriate tools to use under those circumstances?
Okay, do you specifically mean the benthic processes, the plankton processes, like sediment processes?
Yeah, I have to admit I'm a bit challenged in terms of the physical processes that go on with nutrients. That's not my specific area of expertise, but that's also the reason why I kind of try to sound clever and leave the question very generally. But yeah, sedimentation, for example, is a process that can happen on multiple scales of motion, I guess.
Yes, so I just probably explained a little bit for the DELWAC. I think there are multiple groups of sediment. You can specify I think 3 groups or 5 groups. All these can react at different time scales. I think from a few days to a few months to a few years to longer term, 10 years. So when you have all these groups, I think it's just a matter, say if an area hasn't been flooded before, now it's suddenly flooded, then obviously to account for the effect of return nutrients, we need to be able to specify a better initial condition for our model. I think that we'll be able, if we know, if we have some measurements, that would be great. If not, we may have to calibrate the model to account for this effect. And in my opinion, it's just more a matter of being able to specify better initial condition and get a better understanding of the chemical reaction rate. So I'm not an expert in.
I'm sorry, we lost the audio feed. I'm not able to hear anything.
Not me either. This is Josie. I'm not hearing anything. Thank you.
We gotta plug this in, I think. Can you hear it now?
Oh,
Can
you guys hear us now?
Yes, I think so.
Yes. Yes, thank you.
We are running over a little bit. David, did you want to add what you said, I think, online?
I think we're not hearing a sound feed from the room once again.
There's a little battery on that.
Okay.
Are you hearing us
Hang
now?
on. Hang
Yes.
on. Okay, you can hear us now? Okay. Sorry, our battery is dying in our speaker, so we have to plug it in.
Samsey, I think I get the general gist of your question. I know the work that we're doing, at least in the Bay, is not going to try to simulate, for example, decadal timescale changes in sediment conditions, for example, in the Bay. But we know that those things can change and that we need to be considering them. And the approach that I imagine that we're moving towards taking is, instead of actually, let's say, simulating changes in suspended sediment transport and simulating global climate model forcings, and then trying to hopefully bring all those things together and simulate the outcome over decades, we're likely going to instead identify a few likely scenarios of high, low, medium suspended sediments in the water column, changes in temperature or other forcings, and then directly simulating those, as opposed to trying to run all those models and then simulate all of those things over decades within the water quality model.
Okay, thank you. Thank
So
you.
we're gonna do a break. We're gonna resume at, what's the time, right? 3:20. So